going to tell you about this one mastering genetics problem, which is listed below here. It's the Antennapedia with the fruitless. So we are told that we have these two parental chromosomes, A with the dominant, Antennapedia with the dominant gain of function, and a fruitless with a positive. It's wild-type fruitless, so they have normal mating behavior. And then the other parental chromosome is A, that's wild type, Antennapedia that's wild type, and then abnormal fruitless behavior. We're also told that these genes are linked, and they're linked by 15 map units, so that they're going to be 15 map units apart, which, of course, if you remember, goes back to recombination. So the way to figure this out is we figure out the gametes that are formed here, and I'm just going to... we figure out the gametes that are formed. So here we have our two parental chromosomes, and they arise because that's going to equal 85% of our total chromosomes. And then the recombinant chromosomes occur because there's a crossing over event between these two here, okay? So we're going to get A with F plus, and we're going to get AD with F minus, but we're only going to get that a total of 15% of the time. We get each one of those recombinant chromosomes 7.5% of the time, because we need to add those up to get our 15% total recombination, which is equal to 15 map units apart. You then set up the Punnett square as you normally would. So here's my Punnett square, if you can see it, hopefully. And what I've done is I've had the each gamete that I make, that we can make, I either have the percentage of percent that it's going to be based on whether it's a parental chromosome or gamete, or whether it's a recombinant gamete. So you can see that when I have recombinant, I have 7.5%. Here are my two recombinants, and here are my two parentals. And I have the same thing because it's a heterozygous, so it's a dihybrid cross essentially. And then what I do is I figure out the number of individuals that have this particular phenotype, which is what we're supposed to be asking. Legs on the head and normal mating behavior. The genotype is going to be anything that has the Antennapedia dominant allele, and then anything that has one fruitless plus gene, so it has to be wild type. And that's going to be anything that has that is going to be, any genotype that has that is going to give rise to that particular phenotype, legs on head, normal courting behavior. So I've asterisked the ones in the Punnett square that are going to give, that have those genotypes, and then I just multiply the numbers of the predicted times we're going to see that gamete in order to form that particular progeny. So you can see, let's just take an example here. 7.5% of the time I'm getting this wild type chromosome, a recombination event, and 42.5% I have this particular gamete, the dominant allele Antennapedia, and the fruitless allele, that's wild type. So I'm going to multiply 42.5% times 0.75% to give me the frequency of time for the frequency at which I'm going to see that progeny based on the frequency at which each of those alleles are present in the population because of the recombination factor. So I've done the math here for a 42.5 and a 42.5% here, and that's 18.0625% of the time. We're going to get this number, number of progeny that have this genotype. And then this time I'm going to multiply 0.75, because it's this recombinant gamete times the parental gamete, which is going to give me 3.1875. And then lastly, there's another one, which is here when you have two recombinants forming to get the right genotype to give rise to that phenotype, and that's going to be 0.075 times 0.075, and you get 0.6502, I don't know, 6525 or something like that, 0.5625. Anyway, what is it? It's 0.6525. Okay, so then what you're going to do is you're going to add up, you're going to multiply, well, you can add up all of these progeny then with these particular values. You're going to add all of these up, And the way I did it is I just did it this way, where I had, I multiplied 18.1625 times 3, because 3 times I had that 18.56 percentage times. So it's these 3 are going to give me 18% progeny of these 3 right here. I did the same for this. This is multiplied by 4, because I had this potential with these gametes 4 times. I see that in the Punnett square, and then I have this one twice. And then I get this number that is 68.0625, which is equal to the frequency of 0.6806, or something close to that. And so that's what you're going to put in the value, and that's what you get for your answer, and it's correct. And then you do the same thing using the different, the same approach, but you'll look at different genotypes for the other questions. So I hope that helps.